nervous system. But one muscle that we are going to look at this quarter is the muscle that is found in the heart. So does anybody know what the type of muscle tissue found in the heart, what that is called? Cardiac muscle. And then we have another type of muscle tissue, which is involuntary, and that is found in our digestive tract and in our blood vessels and in our bladder, for example. And what type of muscle tissue would that be? Smooth. And then we have the muscle that we think of when we're talking about our biceps brachii or our hamstrings, for example. And that would be, what is that called? Skeletal. Okay. So all of these are specialized cells, specialized tissues that are capable of contraction. And we now know that the way that muscle works is that it pulls to cause the movement instead of pushing. So if you think about your muscles, the ability of me to be able to flex is due to the contraction of my biceps, but my biceps muscle can't push my forearm away. So I have to have an antagonistic muscle, the triceps, which then when it contracts, it pulls my forearm and I can extend my arm. So the contraction has to do with a shortening of the muscle cells. So when we look at the different types, cardiac and smooth are generally said to be involuntary, which means that they are regulated by part of our brain that is not conscious. Whereas the skeletal muscle is voluntary, right? Because we have conscious control over much of our motor movement. Interestingly, the skeletal muscle can also work reflexively. So for example, I am not consciously controlling all the necessary muscle contractions that are keeping me upright, but an unconscious part of my brain can also do that. And so skeletal muscle can also be involuntary, which would be unconscious control. Now, muscle itself has in it muscle fibers that are composed of protein. And does anybody know what those muscle fibers are called? There are two of them that will actually connect to one another and cause the whole cell to constrict. Does anybody know what those muscle fibers are called? So the protein muscle fibers are called actin and myosin. So protein is very abundant within muscular tissue, which is why when we want to eat animals, we generally eat their not their organs, not their livers or their spleen or their stomach. Well, the stomach actually has quite a bit of muscle in it, but we eat their skeletal muscle because it is full of protein. So those protein fibers are called actin and myosin. So if we look at the structure of these types of tissue, they look very different. So if you think about our skeletal muscles are actually composed of very long cells. So in order for these cells to become so long, they actually fuse so that the stem cells that make up your embryonic muscles fuse together and they create these very long cells. And so when we look at skeletal, it is skeletal muscle. So this would be a picture of skeletal muscle, right? It is long, multinucleated, and striated. And so those striations are actually due to the orientation of actin and myosin. The reason why it's multinucleated is that it's caused by a fusion of embryonic stem cells. And it needs to be long so that we can have the contraction of these cells that could cause the contraction of the muscle, the whole muscle. So we're going to spend a lot of time talking about the physiological mechanism of muscle contraction next quarter. So one of the really interesting things about muscle building is that when you build muscle, oftentimes it's not thought to be an increase in the number of cells, but rather the muscle cells themselves get bigger. So it's kind of like fat cells in that our fat cells, when we get fat, they expand, and then when we lose fat, they just shrink up. So when we build muscle, it's believed that the muscle fibers actually increase in their diameter. So the muscle cell will actually increase in its diameter. There is some evidence that suggests that sometimes if you really, really work a muscle, sometimes you can cause tears in the muscle, and then this could cause stem cells to come in and repair. So that tearing of the muscle might actually increase the total number of muscles. So I'm thinking about like bodybuilders when they want to increase muscle mass, they're hoping that they will actually increase the number of muscle fibers or muscle cells they have. Okay, so skeletal muscle is generally connected to bone via what type of connective tissue? So what type of connective tissue would be connecting this muscle to the bone? Tendons, right? Ligaments connect bones to bone. Tendons connect muscle to bone. And that is dense, regular connective tissue. So when we look at cardiac muscle, we see that it is striated. Generally, there is only one nucleus per cell, so it's said to be uninucleated. Oops, uninucleated. So these muscles cells are shorter, and they're striated. And one of the interesting characteristics of them is that they have these structures that separate the cells that are composed of protein, and these are called intercalated discs. So the cardiac muscle cells have to contract and pull one another in order to allow the heart to contract, because the cardiac muscle is not connected to bone, so it can't pull and use the bone as a lever. So it actually kind of pulls against these intercalated discs and has them contract. And one of the really interesting things about cardiac muscle that is unique is it can contract without nervous stimuli. So you can actually take a heart cell out of the heart and put it in a petri dish with nutrients and giving it lots of oxygen and nutrients to survive, and it will contract on its own. So individual cells will contract on their own, and then when they touch one another, they tend to contract together. And so I talked about what type of junctions occur between cardiac muscle cells that would allow the flow of ions from one cell to the other. Does anybody remember that one? Gap junctions. So gap junctions are pores that exist, and so when the muscle cells touch each other, the electrical current, the flow of charged particles, can actually travel from one cell to the other and can cause them to contract synchronously. And then obviously in our heart, we'll talk about this, but we have some tissue that's kind of located up here in the right atria, which is called our pacemaker. And the pacemaker is what regulates the contraction of the heart. Okay. The last one is the smooth muscle, and smooth muscle is not striated. It still has actin and myosin, but we don't see it. We don't see those structures when we look at them underneath the microscope. And if you look at here, they only have a single nuclei. And they form sheets. So when we look at the in the digestive tract, for example, There are sheets that run circular along, like they would run like this, circular. So we have circular muscle. And then we have longitudinal muscle. So they're running in different orientations. So the circular muscle would actually cause the contraction. So if I'm talking about the small intestine, it would cause the small intestine to become smaller diameter. So it would contract like this. And then longitudinal would actually cause a contraction this way. So it would cause the intestine to shorten and also get bigger inside. And so this alternating contraction of the circular and longitudinal muscle is what is referred to as peristalsis. And peristalsis is really important because this wave of contraction keeps the food moving through your digestive tract. And when you get really, really sick, you have a really bad infection and you're in the hospital, sometimes the peristalsis of your digestive tract will stop. And then the doctors get really concerned about that, and they try to get you up and walking. That's one of the reasons why doctors are always trying to get people to walk, is that by walking, that sometimes restarts the peristalsis in the digestive tract. And so this is actually coordinated internally by what is called the second brain of the digestive tract. So our first brain would be in our head, but we have a lot of nervous tissue associated with our digestive tract, and that's sometimes called the second brain or the gut brain, and it actually regulates peristalsis. And you can disconnect the digestive tract from the brain, and it will still work. So it will still secrete digestive juices and undergo peristalsis, even if it is not being regulated by the brain, but it's still regulated by nervous tissue. So that's the difference between it and cardiac muscle. Any questions about those ideas? So that's the muscle tissue. The last type of tissue is the nervous tissue, and the nervous tissue is used for communication. And if we think about the nervous tissue, it generally has it's fast acting, but it is generally short term. And I'm comparing this to the endocrine system. Okay, so this is compared to hormones. So the nervous system and the hormonal system are very much interlinked, and so they complement one another. But the endocrine system produces what for communication? Hormones. The nervous tissue releases what for communication? Neurotransmitters. So this releases neurotransmitters in the what is called the synapse between two neurons. It could also be between a neuron and another structure like a gland. But generally when we're talking about the nervous tissue communicating, it's between one neuron and the other. So these neurotransmitters are released and then they diffuse across the synapse, and then they can cause something to happen in the next neurotransmitter. So sometimes we talk about neurotransmitters as being excitatory, right? So an excitatory neurotransmitter would be like dopamine. So dopamine causes more action potentials or more stimuli to be produced in the brain. We also have inhibitory neurotransmitters. And a good example of this is serotonin. So you hear about serotonin a lot because people with anxiety tend to have not enough serotonin in their brain. And so they take what are called serotonin reuptake inhibitors. and that increases the amount of serotonin between their neurons, and so that decreases the electrical activity in their brain so that they're not so anxious or have panic attacks. So like Prozac, Celexa, Zoloft, and those kinds of things. So one of the characteristics of nervous tissue is that these neurons are highly branched. So when we look at the nervous tissue, this would be the cell body, and then we have these branches coming off. And these branches are referred to as the axon. So an axon is the cytoplasmic extension of a neuron. So it is how it connects to other neurons and to muscles. For example, when we stimulate a muscle to contract, the axon of a motor neuron would release a neurotransmitter called acetylcholine. Okay. These axons can be very long. So a nerve is actually a bundle of axons. So one of the craziest things about our nervous tissue is that you have these cytoplasmic extensions of many neurons that reside in your spinal cord. And then the nerves that run out are just these bundles of cytoplasmic extensions. So the longest cell in your body is not skeletal muscle. The longest cell in your body would be that which runs in a nerve that is called the sciatic nerve. So the cell bodies actually reside in your spinal cord, up here actually in your lumbar region, and then they come down, and then they come down the back of your leg and to your toes. And so that is just a bundle of these little cytoplasmic extensions. They're very long. And so nervous tissue, as well as like skeletal muscle tissue, doesn't easily regenerate itself. So we're going to talk about tissue regeneration. So when we damage your spinal cord, or if you damage a nerve, it is not easily regenerated. It's very slow to repair itself. Okay, so that is very, very specialized types of tissue. Okay, so let's talk about tissue repair. So there are three main phases of tissue repair. So the first phase is what is called inflammation. And some of the signs of inflammation are swelling, redness, and heat, and pain. And pain is actually an adaptive feature that allows us to know that our skin has been damaged, right? Or our organs have been damaged. So pain is actually really good because it tells you something is wrong. Okay? These other signs of inflammation are also good as long as they're short term. When you talk about inflammation being bad, it is generally chronic inflammation. So if I have chronic inflammation of my blood vessels serving my heart, that is a really bad thing that kind of predicts that I'm going to have heart disease problems. But in essence, inflammation is actually a good thing. Okay. So the swelling actually has to do with mast cells. And mast cells are specialized white blood cells. And these release histamine. And these cause the capillaries to become leaky. So again, we think of histamine as being a very bad thing. If our throat swells up from a bee sting, that would be very bad because we could die from that. But histamine is actually an important substance in the body. So what happens when this happens is that neutrophils, which are white blood cells, we talked about these are phagocytes. They leave the circulatory system. So they leave circulatory system and they travel out into the tissues. Okay. and they essentially can eat bacteria and debris. So like if you have damaged tissue, or if you've got like dirt in your wounds, right? It might go in there and it would eat some of the debris. So the neutrophils wouldn't be able to get out of the capillaries. Normally, they're not able to get out of the capillaries except because of the histamine being released, okay? Another way that they actually could get out of the capillaries is if you're bleeding, right? So we also have platelets. So platelets are pieces of white blood cells that help to form clot. So these are involved in clot formation. We also have clotting factors. So we have different clotting factors in our blood that tell or allow the blood to clot when appropriate. And again, you don't want your blood to clot when it's not appropriate, right? So like if you sit too long and your blood starts to clot in your legs, that's a really bad thing, right? So clotting factors need to be highly regulated. And if you have a mutation in a gene that codes for a clotting factor, it could be that your blood clots too much, right? And then you have to take, what would you take if your blood clots too much? Blood thinners, right? And then if you're hemophilia, that means your blood doesn't clot enough, and then you have a bleeding disorder, right? So this balance between when your blood should clot and when your blood should not clot is very delicate and is regulated by homeostasis. Okay. So if we look at a diagram that shows this first part, inflammation. So this is the blood cell. This is the neutrophil, and the neutrophils are going out. So notice the neutrophils are nucleated cells that go out into the tissue and help to repair the damage. We also have those inflammatory chemicals. In this case, would be the histamine, for example. They could be causing inflammation to occur. And we have pain. And then notice that we have the clot in the wound right there. So the scab is actually just the dried clot. Okay, so the second thing that has to happen is we have what is called organization. And so this is the second part of tissue repair. And so one of the cells that is present in the tissue are what are called fibroblasts. And these produce collagen fibers. The fibers extend across the gap of the wound. And this pulls the tissues together. Right. So you do have some ability to heal a cut in the body, right? Sometimes that cut could be too deep or too thick, right? And then you get it sutured. Now they're just having everybody super glue their wounds, right? And that's essentially what just helping the fibroblasts, you know, put the wound back together again, right? So if it doesn't heal, you can suture or super glue. Okay. The other thing that happens here is that we get what is called granulation tissues. Or granulation tissue. And this is pink because it has lots of capillaries. And it is easily damaged. So the granulation tissue is what forms underneath your scab. So if you pick the scab too early, you'll see the granulation tissue, right? The granulation tissue will also puff up, like when you wash it. So like if you're in and you're washing and you pull off a scab and you're washing with water, water will move in via osmosis and then it'll kind of puff up, right? And that's the granulation tissue. And then finally, we have regeneration. So we do have some ability for our tissues to regenerate. Our skin regenerates very well. Regeneration sometimes slows down when we get older. And regeneration is the rebuilding of previous tissue. Okay. So for example, your epidermis being able to rebuild itself. Sometimes regeneration doesn't occur, so then we get what is called fibrosis. So fibrosis is scar formation. So sometimes when we grow really fast, or like if females are pregnant, sometimes our skin cannot keep up with the expanding size of our bodies. And then we get what is called stretch marks. And so the stretch marks are where you're underneath your pelvis is growing in females, and it leaves behind pink stripes, right? That would be the tearing of the skin. And then you get fibrosis, you get scar formation. So they start out red, but then pretty soon they just turn to kind of a white color. Right? You can also get scar formation in your organs. So you can get fibrosis. And so people that have had, for example, heart surgery, sometimes they get a lot of scar tissue built up around their heart. And then that prevents the heart from being able to pump efficiently. And that could actually lead to further heart disease. So we kind of want to avoid scar formation because this can lead to adhesions. So this is where the tissue, right, forms connections where it shouldn't be. So tissue forms connections with structures. So for example, my niece was born very premature and part of her digestive tract actually died. And so they had to go in and remove part of her large intestine when she was a baby. But then as she got older, she got adhesions. And so what was happening was her large intestine was actually kind of adhering to itself and to the surrounding structures. And so then they have to go in and they have to remove the adhesions, right? So we want to try, the surgeons try to avoid at all costs, try to figure out how to prevent adhesions from happening. So sometimes when you get a surgery, they tell you to massage the scar tissue so that if you're cutting through muscle, they don't want the muscle to adhere to the skin unnaturally to form scar tissues, and so they massage it to try to break up the adhesions. Okay. So scars are white in color, and why are they white in color? Oops. Collagen fibers, yes. So this would be the fibroblasts. These are large macrophages that go in and also help to clean up the debris, right? This is the connections between collagen fibers, right? This is the regenerating epithelium. And then we get underneath, oftentimes we have scars underneath in the dermis that we might not see so much above in the epithelium. Sometimes the scars can last a lifetime though. Are there any questions about that idea? Tissue repair? Okay, so when we look at the membranes in the body, there are three types. We have the cutaneous membrane. So what's the example of a cutaneous membrane? What does subcutaneous mean? Underneath the skin. So this is the skin, right? This is a dry membrane. The cutaneous membrane actually helps to prevent water loss. And it does this by producing a glycolipid. So lipids are water-fearing or hydrophobic, right? And so they repel water. And so we are slightly water repellent, right? Our skin is, because it has that glycolipids in it. We don't actually gain water too much across the surface of our skin either. Okay. We also have what are called mucous membranes. So where would be an example where we would find mucous membranes in our body? In nose. So in the respiratory tract. Where else? That would be, yeah. How about digestive? So the mucous membranes are moist and they produce mucus. And what type of cell produces mucus? What is it called? Starts with a G.
Goblet cells.
Goblet cells. So goblet cells they're in the epithelial tissue and they produce the mucin, which then mixed with water produces mucus. Okay. So this would be goblet cells producing mucin. Okay. Then we have a membrane called a serous membrane. And we're going to talk quite a bit about serous membranes this quarter, because this is the type of membrane that you have in your joints. So this is in the joints, and the serous membrane secretes a slippery substance. So this is lubrication. So slippery serous fluid. So the serous fluid is produced when we move our joints, for example, and it actually, as it warms up, it becomes more fluid. So when your joints are cold, then your fluid is is thicker. So it's more viscous when you're cold. And then when you warm up your joints, it becomes more fluid. So this is found in the joints. But it is also found in our body cavities. So for example, it is found in the pericardium. Okay, so that actually we'll put pericardial cavity. Right? So we have a series that says pericardial, D-I-A-L. We have two membranes. It's a double layered membrane, and then we have a space. And that pericardial cavity is filled with serous fluid, so that when our heart moves in our body, it doesn't have friction against the other organs. And so it moves in a friction-free way as the heart beats. Another example is in the pleural cavity. So what is the pleural cavity around? What organ? Lungs. Okay, so again, if you lose fluid or if you get too much fluid, then your lungs aren't able to move properly within their cavity. And then our abdominal cavity is lined by the peritoneal. Okay, so this is the main abdominal. Okay, so if you cut yourself open, you would have a main abdominal cavity, and it would be filled with fluid. It would be filled with serous fluid. So if we look at some pictures of these in your book, so this is just showing the two layers, the parietal and visceral peritoneum, but between those two layers would be the cavity. Okay? The mucous membranes in the respiratory system, the pleural and cardio cavity. Okay, so those are examples of serous membrane. Okay, so we're going to talk a little bit about these embryonic tissues because we start out as 3 tissue layers as an embryo. And so the tissue layers are first described as where they are located. So for example, the ectoderm is on the outside. In all of the diagrams and books, you'll see that this is blue in color. So that's just the color that they give it. They give it blue. So the ectoderm is obviously going to become our epidermis, our outer skin, but it also becomes our nervous system. So all our nervous tissue is derived from the cells that are on the outside of the embryo. And what happens is, you don't need to draw this, but what happens is you get this invagination that kind of looks like this on the outside of our embryo. And then this invagination turns into a neural tube. So then this becomes, this is a cross section. So this neural tube would become your spinal cord, and it would also become your brain. Okay, but it's an invagination. So you talk about the neural plate and then the formation of the neural tube, and that would be the central nervous system. Okay. The mesoderm means middle. And this becomes the muscles and the connective tissue. It is always diagrammed as red in color. And then finally, endo means inside, endoderm. Okay, so that's the inner layer. And this is going to become the lining of the digestive tract, for example. And it is always diagrammed as yellow. So we start out as a single cell, then we become this multicellular embryo that's like an embryonic disc. And then those cells migrate, and then they specialize by regulating their genes. Some genes are turned on in some cells, some genes are turned off in others. And then they become the very specialized types of tissue that we see in our body. So in your book, at the very end of chapter 4, they show this early embryonic disc. And so this is the nervous tissue. This is your neural plate. And this is where you get the neural groove, and you're going to form the neural tube. The red would be the muscle and connective tissue. And then the inner lining of the digestive tract is the endoderm. So this is very early on. Do we see, 16 days? Do we see, what is that, 2.5 weeks, 2 weeks? We start to see the specialization of the cells. Okay, so we are going to now move on to the integumentary system. So what the integumentary system consists of is the skin glands, the hair, which is a derivative of the skin, and nails, which is also derived from skin. So when we look at the skin, we have, or we can talk about its variety of functions. Its first function is pretty obvious in that it is protection. So our skin protects us from microorganisms getting into it. It also prevents water loss. So these are the two greatest dangers if you burn your skin. If you have skin burns over a good proportion of your body, is that you'll get infections and then you'll lose too much water and you'll get dehydrated. And so that's why it's very dangerous to have large surface area of your body burned. Okay. The other thing is it's involved in thermoregulation. So how do you think it regulates body temperature? Sweat. It is also associated with sensation. So tactile senses. Which is important. And it also has a function in excretion. What does excretion mean? What are we getting rid of when we get rid of? What kind of waste? Nitrogenous waste. Now, obviously, the main organ for excretion is not the skin, but rather it is the kidneys. But sometimes in our sweat glands, we also secrete urea. So this is why when you exercise and you get really sweaty and then you put your gym clothes like on a pile in your bedroom and you leave them there for a week, right? This can be converted to ammonia by bacteria. Okay. So bacteria can take that urea and use it, break it down, produce energy from it, and then they excrete ammonia, and that's why your clothes smell really bad. And then you would use vinegar to counteract ammonia because ammonia is base and vinegar is an acid, and so it would help to neutralize and get rid of the smell. Is there anything else that I forgot here? Oh, I did. Metabolism. So metabolism is the breaking down of molecules or the construction. So it could be the synthesis of molecules. So what substance is synthesized by the skin? Anybody know? Vitamin D. So cholesterol in the skin can be converted to vitamin D with sunlight. So I like to think of this as the animal version of photosynthesis, right? We can't produce sugars, but we can produce vitamin D, right? So we use light energy to do that. So this is why when people move up north and away from the sun, they don't get enough sunshine on their skin because they're bundled up because it's cold. They can have a deficiency in vitamin D, which limits their ability to absorb calcium. And so what that would do is that would make a person more prone to bone deformations like rickets. And so we supplement vitamin D, we put it in with milk. So almost all the milks that you see when you buy it from the grocery store have had vitamin D added to them as a supplement. Now, one thing that the skin protects us against is sunlight. So it protects against ultraviolet radiation. That is sunlight. How does our skin protect us from UV radiation? UV radiation could cause mutations in our genetic material, which could potentially become, make our cells become cancerous, and so we could get skin cancer. So how does our skin protect us against UV radiation? You know? Well, sunburn, so the cells will die if they're overexposed, but before you get a sunburn, what do you hope to get? A tan, right? So we have melanocytes. And these melanocytes produce pigment. Right? And this pigment is called melanin. So if you get a tan, you're going to be more protected against the UV radiation. But also, if you are naturally darker in skin color, you're going to be more protected against the ultraviolet radiation as well. So I'm going to show you a little video that talks about skin coloration and its significance. And in this video, they have little places where you stop and you like take a little quiz. So, and they show where that would be on the video. So this video, I think, is about 15 minutes long. Is that right? 18 minutes long. Okay, I want it to be bigger than that. Let's see. Look bigger. Huh. Let's see.
Human brains are gray. Human blood is red. Our bones are off-white.
Let me just see if I can get this to be a little bit bigger. Maybe it's because I'm trying to use the version. Is that bigger? Oh well.
Doesn't matter where you're born or to whom. But human skin is different. Some of us have rich dark brown skin. Some of us have pinkish white skin. Most of us are somewhere in between. For the longest time, why this variation exists was a real scientific mystery. That opened the door for some to invest this biological trait with moral value, and then use that to justify the suffering of others. But biological traits aren't good or bad. They're features that have evolved because they enhance an organism's odds of surviving and passing on its genes. Like other animal traits, the sepia rainbow of human skin color evolved through natural selection. Now, thanks to advances in anthropology and genetics, exactly how and why it did is no longer a mystery. Biological anthropologists like myself spend our lives studying how humans evolved and why we differ from one another physically. Our skin provides one of the most visible markers of human variability. It's something that sets us apart from our closest animal relatives. Under their dark fur, chimpanzees have pale skin, and millions of years ago, that was probably also the case for the primates that were our common ancestors. So where did humanity's range of skin colors come from? From physics, we know that the color of any object comes from the wavelengths of light that it reflects back to an observer's eye. We see leaves as green because they reflect back the wavelengths our eyes see as green. Absorbing the wavelengths we see as other colors like blue or red. In humans, different wavelengths of light are reflected or absorbed by a pigment in the top layer of our skin. That pigment's called melanin. It sits inside what look like tiny grains, the melanosomes, that are produced by cells called melanocytes. Our individual genetic inheritance determines the type of melanin inside our melanosomes. The reddish-yellow pheomelanin is more abundant in lightly pigmented people. More darkly pigmented people have more of the brown-black eumelanin. And the more eumelanin, the darker the skin. Melanin also colors human and animal hair and the feathers of many birds. Interestingly, the wavelengths of light that melanin reflects are far less important biologically than the ones it actually absorbs. And of the ones it absorbs, the ones that are the most important are those that we can't even see. Much of the light given off by the sun is invisible to our eyes. Some of that is what's called ultraviolet radiation, which is highly energetic. So much so, it can actually penetrate living cells. When it does, it can wreak havoc within them. It can even cause mutations in skin cell DNA. What stands between us and that threat is the melanin in our skin.
Melanin is kind of like the sensor. It's kind of like a guardian molecule, and its main job is protection.
For instance, it protects skin cell DNA by forming what are called supranuclear caps and absorbing UV.
They're like little parasols around the nucleus, and UV cannot penetrate these to go and attack the DNA.
That's just one of the things molecular biologist Zalfa Abdelmalek finds remarkable about melanin. Another is the broad range of benefits it provides a diverse collection of species.
We know that melanin in our vertebrates is important for regulating body temperature. It can also give animals camouflage and allows them to recognize other members of the species to propagate the species.
Okay, so which of the following statements about melanin is most accurate? Having melanin is one of the distinguishing features of humans compared to other animals. Is that correct? No. Everyone has the same type of melanin, but the amount varies from person to person. No, because there's the pheomelanin and there's the eumelanin. The more brown-black melanin an individual has, the darker their skin. That much is correct, I think. And the more brown-black melanin an individual has in their skin cells, the more likely they are to get mutations. No. Okay. So the correct answer would be C. See if we're right. Yes. Okay. Which of the following statements are true concerning how light interacts with pigments in a red tomato? The pigments absorb blue light. The pigments absorb red light. The pigments likely protect the cells in the tomato from DNA damage. The pigments reflect green light. Fruit generally is colored because they're trying to attract birds, probably, so that they could come in and get the fruit and spread the seeds. So I'm thinking that it is A, but I'm not sure. What does everybody else think? Yes. Okay.
In humans, one of melanin's functions is clearly to protect cells from UV damage. As we evolved, we lost hair and increased melanin production in our skin. So, is there a connection between the intensity of UV radiation and skin color?
Hi, Tess.
I first became fascinated with UV and skin color in the 1990s. But as I searched for information about the global distribution of solar UV, I discovered the available data was in fact quite spotty. I began casting a wider net, and almost by accident, found exactly the raw data needed to fix that. It hadn't been collected by anyone interested in my questions, but rather by NASA. In the 1980s, concern about the health risks posed by the depletion of UV-blocking atmospheric ozone led NASA to take millions of UV measurements from space. I asked NASA to send me the data, and then asked my geographer husband, George Chaplin, to try to visualize it. It turned out to be a bigger request than I'd realized, but he found a way to turn all those data points into a map that showed for the very first time exactly how UV exposure varies throughout the world. Most striking was the clear gradient between the equator and the poles, which was interrupted only in places where altitude increased UV exposure.
This is actually in the Tibetan plateau.
And persistent cloud cover decreased it.
Our basin, so it's full of humidity and moisture, which is blocking the UV.
Solar energy is a fundamental attribute of any environment. And it's a well-established fact that organisms living at different latitudes adapt in some way to their local solar conditions. To see how closely human skin color correlates with UV exposure, I collected skin pigment measurements made by anthropologists studying indigenous peoples. For many years, anthropologists have faced the challenge of how to accurately measure skin color. We now use this little device called a reflectometer. Basically, it sends out light of specific colors, and then it measures the amount of light that is reflected back. This tells us what color Tess's skin is, and we can then compare this to people all over the world. George then created a second map using measured skin colors and environmental data. It showed UV intensity does indeed predict skin color. Wherever UV is strong, skin is dark, like it is near the equator or at high altitude. At the poles, the skin of indigenous people is almost always lighter. That suggests that variation in human skin melanin production arose as different populations adapted biologically to different solar conditions around the world.
Okay, so in which of the following areas of the globe would you expect to find relatively high amounts of UV radiation? Okay. Area with a large amount of persistent cloud cover? Nope. Areas at high elevation? Yes, so at the top of the mountains, they would. At areas at a latitude near the equator? Yes. Areas at longitudes in the Western Hemisphere, so that would be like, not latitudes, but it's going this way from pole to pole. So what do we think it is? C and C. Oh. Yes. Okay. Which of the following statements apply to the variation in human skin color? Human skin and the skin of our closest living primate relative exhibit a similar range in color variability. Do monkeys have, or chimpanzees or gorillas have variation in their skin color? No. Okay. Human skin color variation is primarily determined by the type and amount of melanin pigment in the skin. Yes. The variation in human skin color cannot be quantified. No, that's not correct. The variation in human skin color correlates with the intensity of UV radiation around the world. Okay.
As we've noted, our early ancestors probably had full body hair covering pale skin, just like other primates. So when did the darker shades of human skin begin to evolve? DNA sequencing has made it possible to find evidence that can help answer that question. Rick Kittles is a geneticist who's skilled at deciphering such clues.
Whenever a species undergoes some form of selection, some form of natural selection, evidence of that selection is found in the genome. And so as geneticists, we get really excited when we explore the genome for these signatures. One way in which that's done is by sampling worldwide populations and looking throughout the genome at variation and comparing across populations. And it's a very exciting process. I feel like a detective when I go through that process.
One of the many genes that genetic detectives have linked to human pigmentation is called MC1R. Sampling from around the world indicates there's a fair amount of variation in the DNA sequence of that gene, but not from every corner of the globe.
When we look at MC1R within African populations, we don't see a lot of diversity. And the particular allele that they have in those African populations is the one that codes for darker skin. MC1R codes for a protein which is involved in the switch from the production of pheomelanin to eumelanin. And we know pheomelanin is the reddish yellow pigment, and then the eumelanin is the brown black pigment.
The absence of MC1R diversity in African populations indicates that, in that part of the world, there is strong negative selection against any alleles that would alter dark skin. And how long has this allele been fixed in African populations? Other genetic studies have calculated that it has been as much as 1.2 million years. Since our species evolved in equatorial Africa, it's reasonable to conclude that by that time, all humans were dark-skinned. The fossil record supports what we glean from genetic evidence. But here's where we confront what was, for me, the heart of the mystery. The evolution of dark skin in humans suggests that under strong UV light, that trait provided a survival advantage. So what exactly was that advantage? It's certainly true UV damage to skin cell DNA can lead to cancer, and skin cancer can be fatal. For a long time, that seemed the likeliest explanation, except skin cancer generally develops after a person's peak reproductive years. For that reason, though it might cut your life short, it's unlikely to affect your ability to pass on your genes. As I was struggling to conceive of an alternative explanation, I happened to attend a lecture on severe birth defects. That talk was about a research project that had found evidence that certain birth defects are far more common among pregnant women with diets deficient in a B vitamin called folate. Only weeks before, I'd come across a paper that described how strong sunlight breaks down folate circulating in skin blood vessels. Here was a direct link between UV radiation, skin color, and reproductive success. It was a small eureka moment for me. In the years since, we've learned that folate is not only essential for normal embryonic development, it's even needed for healthy sperm production in males. Folate is biological gold. It is an essential nutrient, and it needs to be protected from UV radiation as it circulates in the blood vessels in the skin. That is what melanin does. I felt I was halfway home on my quest to understand human skin color variation. But a big question remained. Why aren't we all dark-skinned? It turns out there's another side to our relationship with UV light. UV light is not all bad. In fact, the small portion of it known as UVB is critical for the synthesis in our bodies of vitamin D, a process that starts in the skin. Without vitamin D, humans cannot absorb calcium from our diet to build our bones and for a healthy immune system. Back when all our ancestors lived close to the equator, there was no problem getting enough UVB through dark skin to make the vitamin D needed. But then some populations started moving north, where the UV striking Earth's surface is much weaker. In northern latitudes, dark skin makes it hard to produce the vitamin D that human bodies really need. The consequences of vitamin D deficiency include rickets, a bone development disease that can cripple the young. In higher latitudes with less UV, the selective pressure on MC1R that produced dark skin in our ancient ancestors began to abate.
When we look at the early movement out of Africa, when that constraint was relaxed, we then see a plethora of variation.
In European and Asian populations, geneticists have discovered greater variation in the MC1R gene, but less variation in several other genes, ones associated with lighter skin types.
Different environments led to other genes being selected for and being important for those populations in terms of skin color.
Selection for light skin gene variants occurred multiple times in different groups around the world, some of it in just the last 10,000 years. Support for the idea that the UV vitamin D connection helped drive the evolution of paler skin comes from the fact that indigenous peoples with diets rich in this essential vitamin have dark pigmentation.
Okay, darker skin is more prevalent in high UV areas. Dr. Jablonski proposed a hypothesis to explain the selective pressure for darker skin in these environments. On what evidence did she use or to base this hypothesis? People of African descent have a very similar version of the MC1R gene, indicating what the selective pressure was for darker skin. In general, people of African descent have less folate, a nutrient important for human reproduction, circulating in their blood. No, that's not correct. A large amount of brown, black melanin in darkly pigmented skin protects circulating folate from being destroyed by UV radiation. That, I think, is the correct one. The melanin in darkly pigmented skin protects DNA from UV radiation, which can cause skin cancer. Okay, so this folate is also sometimes referred to as folic acid. And so if I was pregnant and I was light-skinned in a dark environment, then my folate would be broken down, and then perhaps my embryo would not develop properly and would have birth defects. So what type of birth defects does anybody know that's related to lack of folic acid? Nervous system, right? So like neural tube defects, like when babies are born without their nervous system developed, or they have a spina bifida, which is their nervous system is outside of the vertebrae. So that's why today, when you're pregnant, the doctor gives you what? Or tells you you must take prenatal vitamins. So they even recommend that you don't wait until you're pregnant, that if you're trying to get pregnant, you take prenatal vitamins so that there's folate in them. Okay? So the folic acid can help with the development, normal development of the nervous system. Okay. Human populations in low UV environments tend to have lighter skin tones. One explanation is that the selective pressure for dark skin decreases as UV intensity decreases. At the same time, there's selection for lighter skin to absorb more UV radiation, which is needed for vitamin D production. What evidence supports the vitamin D hypothesis for the lighter skin tones? In general, people with lighter skin tend to be at a higher risk of vitamin D deficiency. That's not true, right? It would be dark. So if you were a dark-skinned individual and you lived up north, sometimes that would be a problem unless you ate the right food, right? So I think the correct answer would be some indigenous peoples living at high latitudes have darker skin, but they tend to eat foods rich in vitamin D. Correct. I don't know if we're going to finish. We'll just take a couple more minutes.
The tension between these two aspects of our biological inheritance, on the one hand, the need to protect ourselves from most ultraviolet radiation, and on the other, the need to use some ultraviolet radiation for our own benefit. These forces drove the evolution of the wonderful variation in human skin color that we see around us today. It's the legacy of an evolutionary balancing act necessitated by the different environmental conditions people have faced around the globe. The thing is, where once human migrations took many generations, we now move about the planet at the speed of sound. That means increasing numbers of us have pigmentation that's not a good match with where we live.
People with fair skin and red hair, your phenotype is telling you you have a high risk of skin cancer if you're out in the sun. If you're a dark-skinned individual, living, for example, in Scandinavia or in Minnesota, you're not going to have optimal exposure to UV for optimal vitamin D synthesis, and you need to take supplements.
We now know that we need to make cultural adaptations like these to stay healthy, but that's not all we've learned.
Okay, so we'll stop there. Okay, so I will see you on Wednesday, and remember that there is a quiz.